There are a number of named lectures which are very prestigious at the EASD. One of the most prestigious is the Minkowski lecture, and I'm very pleased that we have the giver of this year's named lecture is Fredrik Bäckhed from Sweden. Now you're talking about the gut microbiome. Gut microbiome and diabetes, what on earth have they got to do with each other?
Oh, actually, I think they have a lot of things together. The thing we need to figure out right now is if it's just causal or associations. So what we know today is that there's an altered microbiome in subjects with type 2 diabetes. The question is whether it actually is contributing to the disease or it's just a marker of the disease. And I think that the more data we generate is probably a bit of both, but separating those two out from each other will be imperative to be able to use these as signals for the disease.
So just back up a bit. Gut microbiome, I mean, we have 10 times more bacterial cells than we have human cells in our body, extraordinary, much more DNA, bacterial DNA than our own DNA. And we know that sort of variations in the gut microbiota are associated with obesity. Just take us through the links that lead to diabetes.
Well, you're absolutely right. We have more bacterial cells than human cells, and it's estimated that we maybe have up to 100 to even 1,000-fold more bacterial genes than we have human genes in the body. And these will, of course, also change depending on what we eat. And I think that's one of the confounders that we need to take better into account when we do these kind of associations. And you're right that there is a signal in obesity in the microbiome, but it seems to be hard to replicate it. And I think that's partly due to that the studies have been relatively small. And so the common theme in those studies, at least, is that the obese individual seems to have a lower diversity, which means that they have fewer bacterial species in the microbiome. But it hasn't been ruled out if this is a consequence, if the obese individuals eat differently, i.e. less fiber, for example. But I still think that there may be something there, but it will be hard to do interventions, I think, because I think there's many other environmental factors, such as diet and physical activity, that will drive the obesity epidemic. So what our take on it is that why does not all obese individuals go on and develop type 2 diabetes? It's about 40%, at least in Europe, that seem to go on and develop type 2 diabetes. So could there be a microbiome signal that is sort of a driving factor for that transition? And what we start to work towards a hypothesis right now is that diabetes is also a multifaceted disease, so you can get diabetes due to many different reasons. So I think we need to be better at stratifying both the patients on the clinical signs as well as the microbiome. And if we do that, we'll probably find subpopulations where we can have a much stronger association between given bacteria and the subtype of type 2 diabetes. And then we could potentially start to do much more sophisticated interventions.
And what are the big questions, though, that we still need to ask in this area?
I mean, what we see a quite clear effect of the microbiome in cross-sectional studies of type 2 diabetes, which means that you have fewer of the butyrate-producing bacteria. That seems to be good for you in many different diseases. But we don't know yet if it's a cause to the disease or just reflecting the disease. So what we would need is prospective studies, where we also look much more in depth into pre-diabetes and see if we see these changes before. And then to see how we can modulate the abundance of them in humans to see if we can modulate the disease outcome. Right now we have nice causality in animal models, but there's very few causal studies in humans. There's one from Max Nudorp's lab a few years back, which seems plausible that there is a shift in the microbiome that at least can contribute to some of the diabetes phenotypes that we see today in humans.
Now, we know that people with diabetes are particularly prone to thrush infections, candidiasis. Is that a consequence of an altered microbiome?
I would say that's a very likely explanation to it. And I think that those are the questions we need to to address much more as well, if there's a diabetes complications that can be mediated by the microbiome. Because it seems like what the altered microbiome reflects is that you have less butyrate producing bacteria, but you also have signs of oxidative stress and more potential pathogenic bacteria. So those may contribute to a lot of the complications, if not to the disease itself. But those are exactly the questions that we would like to delve in much deeper now, when we have sort of established that there is an altered microbiome in type 2 diabetes. The other question I think which will really need to be addressed is by which mechanisms the bacteria or the microbiome can contribute to the disease, if it does so. And then we are focusing a lot on metabolites that are being produced by the microbiome, to see whether they can affect insulin signaling in specific cell types and as such being contributors to the disease.
And we already know, I mean, we see it in many of the congresses that are going on at the moment in diverse medical disciplines, of the influence of the microbiome in those disciplines, and skin and lung and even neuroscience in terms of the brain. Is the microbiome having an effect everywhere?
That's my. I'm the first to start to wave my hands and say that we have to be cautious, and that is that most of the studies are relatively small, and they are confounded by drugs. Or medications. So we don't know if we actually have a true microbiome signal that contributes to the disease, or just reflecting the disease or the treatments of the disease. And I think that's true for most diseases. We are working really hard right now in the field of diabetes to sort of curate the data. So we are correcting for medication. Metformin, we know, has a huge impact on the microbiome. But similarly, I think we have to do in all kinds of fields, because if you just take 50 patients and 50 controls, I would be more surprised if you did not find a difference. The question
there are extraordinary differences between individuals, let alone.
But what I think is failing in many studies is to see if it replicates in an independent cohort.
Yeah.
When you've done that, I start to believe in it. Then the next question is whether it actually is a disease modifier or can contribute to the disease by doing prospective studies or animal experiments to look for causality. The causality that is done today is mainly done in animals. So I think we have a long way to go in terms of really pinpointing how important the microbiome is. I think it is important, but it is equally important for all kinds of diseases, or it's more important for specific diseases, such as, I hope, type 2 diabetes.
And I suspect that we might have to develop a whole new methodology for dealing with the microbiome and investigating it, because you can't have controls because of that great inter-individual variation.
And I think that what at least our group is focusing on a lot right now, and I know many others do as well, is that instead of looking at the bacteria per se, because they may have functions that can be performed by other similar bacteria. So maybe we shouldn't look at the bacteria, but at the resulting metabolites that they produce. And we can maybe do plasma samples instead and look at those as a result of the shift in microbiome.
And the other thing is that we are obsessed with the microbiome with bacteria, but of course the microbiome is much more than just bacteria. It's also viruses, it's also fungi as well. And those we kind of ignore.
Yeah, and the reason for why we do that is because we don't have the same technology platforms to analyze those. I know that there's a lot of interest in the viriome, so the viruses, and many of these viruses are called phages and infect the bacteria. So that interplay will be extremely important, and I've seen estimates that there may be 1,000-fold more viruses compared to bacteria in the gut. So but then you have to develop first, I think, another suite of tools to do this. And so far, it's only a few specialized labs that seems to be capable of doing analyzing the viriome in great detail. The same thing with the fungi.
There is, though, now the equivalent of the Human Genome Project, but the Human Microbiome Project. How is that going to help?
I think what it primarily will help with is sort of defining what's a healthy microbiome look like? What is the variation we see in a normal population? I shouldn't say healthy because it's of course hard to say, but in a normal population, what are the variations we can expect to see? And also to see what is the normal variation within you and me in our microbiome over a year's time, I think would be extremely important to say, because the variation we see between individuals may just as well be due to variation within an individual.
Yes.
So there's a lot of these questions that we need to start to addressing now, and then we've sort of gone through the 15-year, I think, of a hype, and we are, excited about it. But now we have to stand up to the test and see whether it will still be there in 5 years when we have done the replication cohorts, when we've done the prospective cohorts, when we have really validated all the methodology, when we have started to get into the mechanisms. And I think in order for you and I and the public to care about this, we have to be able to show some kind of a treatment effect based on the microbiome, or at least stratify subjects into different treatments based on the microbiome. I think we are on the verge to that process, but that will of course take quite a bit of time. But I think that's what we should put a lot of our emphasis on right now.
Well, thank you very much for joining us. I think the message here is watch this space. I think the microbiome continues to intrigue, and I think that in the next 5 to 10 years we will see some extraordinary results. Thank you for watching us, and you'll find the whole of the Minkowski lecture on EASD.org.